It's wonderful to have you here with us today. Get ready as we explore some of the most fundamental and fascinating processes that keep our cells energized and our bodies functioning. Welcome to our channel, MooTechie, exploring metabolic pathways, TCA cycle, glycogen synthesis, glycogen degradation, and transaminase reactions, cellular respiration, energy metabolism, biochemical pathways. Now, let's move on to an overview of these vital metabolic pathways. The TCA cycle is the central hub for energy production from carbohydrates, fats, and proteins. Glycogen synthesis is the storage pathway for excess glucose in the liver and muscle. Glycogen degradation allows for rapid glucose mobilization during fasting or exercise. And transaminase reactions involve amino group transfer between amino acids and keto acids. These pathways form an interconnected network maintaining cellular energy homeostasis. The slide also shows a detailed diagram mapping out these connections. We see key molecules like glycogen, glucose, glucose-6-phosphate, pyruvate, and oxaloacetate, with arrows illustrating how carbohydrate, fat, and protein metabolism intersect. Important enzymes like pyruvate dehydrogenase and lactate dehydrogenase are noted, showing the flow from substrates like fatty acids and amino acids through various pathways, including gluconeogenesis, all feeding into or out of the central TCA cycle. The diagram highlights the beautiful complexity of our metabolic landscape. Let us now look closely at the TCA cycle, the central hub of cellular metabolism. First, its location. It occurs in the mitochondrial matrix in eukaryotes and in the cytoplasm in prokaryotes. The key enzymes include citrate synthase, isocitrate dehydrogenase, and alpha-ketogluteric dehydrogenase. The products are 3 NADH, 1 FADH2, 1 ATP or GTP, and 2 CO2 per acetyl-CoA molecule that enters. Its regulation is controlled by the cell's energy status, specifically the ATP to ADP ratio, the NADH to NAD+ ratio, and substrate availability. And its significance is profound. It is the final common pathway for the oxidation of carbohydrates, fats, and proteins. Next, we explore glycogen synthesis, the process of storing excess glucose. This occurs primarily in the liver and skeletal muscle. The key enzymes involved are glycogenin, which acts as a primer, UDP glucose pyrophosphorylase, and glycogen synthase. The pathway flows from glucose to glucose-6-P, to glucose-1-P, to UDP glucose, and finally into glycogen. Regulation is key. Insulin activates glycogen synthase by promoting its dephosphorylation, while glucose-6-P acts as an allosteric activator. This process is significant because it maintains blood glucose levels via the liver and provides a rapid energy source for muscle. Following this, let's understand glycogen degradation, which is all about mobilizing that stored energy. It takes place in the cytosol of liver and skeletal muscle cells. Key enzymes here are glycogen phosphorylase, alpha-1-4 to alpha-1-4 glucan transferase, and amylo 1-6 glucosidase. The pathway breaks glycogen down into glucose-1-P, then to glucose-6-P, and finally to free glucose in the liver or pyruvate in muscle for energy. It's regulated by activation through phosphorylation, triggered by glucogen in the liver and epinephrine in muscle. AMP also acts as an allosteric activator in muscle. This rapid glucose release is crucial for maintaining blood sugar and providing immediate energy during activity. Moving forward, we'll see the role of transaminase reactions in amino acid group transfer. The mechanism involves the transfer of an amino group, that's NH2, from an amino acid to a keto acid. Key enzymes are ALT, or alanine transaminase, and AST, aspartate transaminase, among others. These reactions require a cofactor called pyroxidol phosphate, or PLP, which is a derivative of vitamin B6. Their metabolic role is critical. They link amino acid metabolism to the TCA cycle via alpha-ketoglutarate. And they have major clinical significance. Elevated ALT and AST levels in blood tests are key indicators of liver damage, making them vital diagnostic tools. Coming up, we have our conclusion on these interconnected metabolic pathways. First, energy integration. The TCA cycle serves as the central hub for energy production from all fuel sources: carbohydrates, fats, and proteins. Second, glucose homeostasis. Glycogen synthesis and degradation work together to maintain stable blood glucose levels. Third, nitrogen balance. Transaminase reactions link amino acid metabolism to the TCA cycle, managing nitrogen. And fourth, metabolic flexibility. Cells can seamlessly switch between these pathways based on energy needs and what substrates are available. Now, let's talk about key metabolic crossroads. Glucose-6-phosphate is a major junction, directing traffic towards glycolysis, glycogen synthesis, or the pentose phosphate pathway. And pyruvate is another crucial hub, linking glycolysis to the TCA cycle, gluconeogenesis, and amino acid metabolism. The slide also features a diagram illustrating glycogen synthesis. It shows a glycogen primer with a tyrosine group and a magnesium ion. We see UDP glucose donating a glucosyl unit in an autocatalytic step. The enzyme glycogen synthase then extends the chain using UDP glucose. Finally, the branching enzyme creates the highly branched structure, resulting in a mature glycogen particle containing about 60,000 glucosyl units. Thank you for watching. We hope you enjoyed learning about these essential metabolic pathways: cellular respiration, energy metabolism, biochemical pathways. Thank you. See you in the next class.